Gracias por venir. Voy a comenzar con el aviso de seguridad. En caso de cualquier emergencia, por favor, la mitad del auditorio sale por esta puerta y la otra mitad por la otra puerta. Y las personas que estén sentadas cerca de las puertas nos ayudan a abrir los dos cerrojos para abrir las dos hojas de las puertas. También a partir de este momento les voy a pedir si pueden apagar sus celulares y ponerlos en modo de silencio. Y también que eviten comer cualquier alimento o bebida dentro del auditorio. El día de hoy tengo el placer de presentar al doctor Scott Egan. Él es un invitado del doctor Alejandro Saldívar. Y el doctor Egan trabaja en la Universidad de Rice en Estados Unidos. Él tiene dos afiliaciones, como profesor asistente en el Departamento de Biociencias y también como profesor asociado en el Instituto de Biociencias y Bioingeniería. Él es biólogo, hizo la licenciatura en biología en la Universidad de Texas y después hizo una maestría en la Universidad Estatal de Texas y un doctorado en la Universidad de Vanderbilt. Su línea de investigación es bastante interesante. Se enfoca en estudiar los procesos que promueven y restringen la evolución de nuevas especies y pues varias de sus investigaciones se han enfocado en estudiar las asociaciones que tienen diversos linajes de insectos, parásitos y sus plantas hospederas. Sin embargo, también ha habido varias aportaciones sobre, por ejemplo, plantas invasoras y algo muy interesante, les invito a revisar una de las publicaciones más recientes del Dr. Egan en Biology, creo que es el número 28, es el reporte de una primera planta que parasita a un insecto. Por fin, la revancha de las plantas contra los insectos, por favor, revísenlo, es bastante interesante. Y pues este es, por ejemplo, uno de los cerca de 70 artículos publicados por el doctor, quien además tiene numerosos proyectos de investigación como responsable de la NSF, también del Departamento de Agricultura, y así como otras becas de iniciación de investigadores de la Universidad de Rice. Entonces, damos lugar a la plática del doctor.
Hola, gracias por asistir a mi seminario y gracias al Dr. Saldívar y su laboratorio por recibirme esta semana. Me complace compartir con ustedes el trabajo que estamos haciendo a las Rice University en Houston, Texas, y espero poder aprender más sobre el trabajo que están haciendo aquí en UNAM. Muchas gracias. Pero mi español no muy bien, so I will try to get my talk in English now. Sorry. So most of my work is to link things such as ecology with evolutionary biology. And one of the greatest ways to do that is to think about the role that ecology plays in the evolution of new biological species, which my lab works on. So this process, this evolutionary process, we call speciation. It's a process in which new biological species arise. You can see right here one of the famous drawings by Darwin over 150 years ago. And our lab focuses on how these lineages split through time. And specifically, my lab thinks about a subdiscipline within speciation, which is ecological speciation, which is simply that species evolve ultimately as a consequence of divergent natural selection between environments, between traits, between environments. MacPC. So two quotes. One, from Dubjansky, very famous quote, nothing in biology makes sense except in the light of evolution. So I teach that in my evolutionary biology class at Rice. But from the Grants, who work on the Darwin's finches, nothing in evolutionary biology makes sense except in the light of ecology. I think both of these statements are true, and so my lab intends to link these two forms to address this process. So how do we study speciation? Well, first we think of the ecology of the system as very important. Natural history observations are very important in our lab. We then link that to behaviors, things like mate preference or habitat preference that can generate reproductive barriers between populations. And then we think about how ecology and behavior are linked within the genome, and how the genome may also play an additional role in facilitating or constraining evolution. And all of these subjects lead to the speciation process. So to study speciation, you need to define what you mean by a species, which is challenging. But for us, we think about it in the biological species concept, which is defined as species or groups of interbreeding sets of populations that are isolated or reproductively isolated from other populations. So for us, the study of speciation is the study of how reproductive isolation evolves in nature. So as you know, if you're a student of evolutionary biology, these types of barriers can evolve before mating has occurred or after mating has occurred. So for example, most of my study systems are in insects, so I'll illustrate this with some of our insects. You can have pre-mating barriers such as habitat isolation. If you strongly prefer to only go to one habitat and not another, you might never interact with populations in an alternative environment. For the purposes of this talk, the insects we look at are all herbivorous, plant feeding. So we think of environments as host plant species. Next, if you interact with individuals of the opposite sex, you may choose to only mate with individuals from a certain environment and not from another, exhibiting some traits or adaptations to that environment. Again, reducing gene flow between populations. If you do go on to mate and then generate offspring, sometimes those offspring will not be functional, they'll be sterile or infertile. We call this intrinsic or genetic post-zygotic isolation, where hybrids are less fit than pure crosses. And then even if those hybrids survive, they may be mismatched to either of the parental environments. So we also call this ecological or extrinsic post-zygotic isolation, where the hybrids just don't fit and survive in either environment. So for this talk, I'll be talking mainly about herbivorous insects. They're wonderful systems to study the process of speciation in action. And they're for many reasons. One is their ecological intimacy with the host plants they feed on. So the host plant is the place where they feed, where they lay their eggs, where they find their mates, and where they hide from predators. So this is a very intimate interaction between the host plant, which is the environment, and the populations that feed on it. Second, herbivorous insects exhibit a high degree of specialization. Most insects only feed on a small subset of the available plants in any given environment, so they're a high degree of specialization. And then last, and what got me into studying insects, is their great diversity. Diversity within species and phenotypes, and then the diversity in the number of species on the planet. Something like 10 to 30 million, and we've only described 1 million insofar. So lots more work to do in describing new species. Today I'll focus on two systems. These are three systems I've worked on. They all share a couple of common traits. They're all herbivorous insects. They have this intimate interaction with their host plant. They all come from highly specialized lineages. Most of the organisms in each of these three groups are one species onto one host plant. But these three species that I work on are oligophagous, so they feed on 2, 3, or even 6 different host plants, but they are surrounded by relatives that only feed on one. And then last, for each of these, we have some evidence of reproductive isolation, but it is not complete. So for each of these systems, we've caught speciation in the act. They are not one group of panmictic populations, and they are not completely reproductively isolated yet. They're somewhere along that process, and that allows us to understand the speciation process as it unfolds. So first today I'll talk a little bit about cynipic gall wasp, which is one of the reasons I'm here to collaborate with Alejandro in his lab. I'm going to go out in the field this week to look for gall wasps here. And at the end I'll talk a little bit about some of the genomics and genetics we're working on with Ragalides fruit flies. which are also located here, including in your parking lot. There's a black cherry tree, which is infested with a species of Raglates fruit fly. I probably won't have time to talk about neoclimasis beetles, but we also work on another group of beetles that's adapted to a certain set of host plants. All right, so let's start off with the gall wasps. So gall wasps have amazing biology. Starting off with, they induce tumors on the tree that are made completely of the plant material, but they are induced and controlled by the insect. Second, they have 2 generations per year. A sexual generation of males and females, and then an all-female asexual generation. Each of these 2 generations induces different types of galls on the exact same set of host plants. For this species, Belenacne matridi, the asexual generation, the all-female generation, develops within these spherical leaf galls. These should be coming out right now in the fall. So these are some of the ones we're going to be looking at with Alejandro's lab. That generates this asexual female. She then descends down to the roots of an oak tree and induces this multi-chambered gall within which many chambers exist, and out of that pops either males or females. They mate, go back in the spring onto new leaves, and induce the next generation, completing the life cycle. So they have this amazing biology and life cycle that's going on. At the same time, they're doing all the things I'm interested in. Across the Gulf Coast of the United States, there are 2 sister species of oak. The first and more common is Quercus virginiana, or the southern live oak. In green, it's distributed along the Gulf Coast from South Florida all the way to South Texas, with a couple isolated populations in northern Mexico. Right here in northern Mexico, another live oak species begins called Quercus oleoides, and that's one of the species we're interested in sampling here. But over in the southeastern United States, there is a second species of oak that's adapted to sandy, dry soils. Quercus virginiana is in more mesic, wet soils. And the populations of our gall wasps feed on both of these oak trees, so this is the system we use to understand how adaptation of the gall wasp to these 2 environments, these 2 host plants, promotes the speciation process by generating reproductive isolation between populations. So how do we do this? Well, we do experiments. So one of the experiments we just recently did in our lab is we, along with a huge set of collaborators, grew hundreds of tiny little potted plants, either of Quercus virginiana and Quercus geminata, and we loaded them all into a moving truck and drove across Florida and left them underneath large trees that were infested with our gall wasps. We let the gall wasps descend down onto our potted trees that were about 5 to 8 feet tall, and they induced galls on all our potted trees. So we would take populations on Quercus virginiana, and underneath that tree we would plant some Quercus virginiana as a control, and Quercus geminata, the other environment, to see if they could induce galls and survive in this alternative environment. What did we measure? One of the biggest factors that the insect has to negotiate with when attacking these large host trees is the immune system of the plant. The plant is programmed to kill parasites. It does that by killing all the cells that are in contact with the insect egg when it gets laid onto a new leaf. So right here you can see these small little holes I've highlighted in yellow. That is what we call a hypersensitivity response, a response of the plant immune system to kill off the egg. We measured this immune system response for individual populations of our insects that were on the native species or on the non-native species. And what you can see here is this immune response was much stronger on the non-native species. So this is one way that divergent selection acts between these populations. If individuals get onto the alternative environment, the eggs rarely survive because of this immune response of their host. But notice, it's not 100%. Some do survive. Along these same experiments, we also measured the degree of egg laying per leaf. So the amount of time and energy the females spend on the native versus the non-native is equal. They're laying the same number of eggs. So that's interesting. But the outcome of this immune response is that individuals grow into these galls only on the native tree, and very rarely, 1 in every 200 leaves will have a gall form and an adult come out. So this very strong selection from the immune system against individuals moving between populations we call immigrant inviability. The individuals that move, they can get to the environment, but they can't survive to the next generation. They're effectively inviable. So that's one barrier to gene flow between populations. You can't migrate between them because you cannot survive. Another important trait to think about is the phenology or timing of the host plant. Live oaks, this group of oaks that we study, are called semi-evergreens. They keep their leaves all year, but in the springtime they go through this massive flush of their leaves. They drop all of their old leaves and immediately or simultaneously grow new leaves at the same time. So the buds are a very active area of biology in the springtime, critical to both the plant and any of the insects that need new leaves to grow on. Here you can see one of our females waiting for the buds to break. She just sits there and waits for these things. Sometimes she can actually take her ovipositor and shove it into the bud and find the tissue she needs before the bud is even broken. So it's a very critical timing issue for insects and plants. And what are those critical issues? First, the bud contains the catkins, the male flowering parts of an oak tree. So these buds first pop out catkins, and on the very same new growth, the next thing is leaves. So they're all tied into each other. Interestingly, I can piggyback on a lot of great science done on the plants that has already been done by a lab led by Janine Cavender-Bars at the University of Minnesota. She's been studying these plants for a very long time. And her group hypothesizes that these catkins, the timing that they come out, is critical to reproductive isolation between these two sister species of oaks that I study in Florida. So there's selection so that the catkins of one species come out later so that you don't generate maladapted hybrids of the oak trees. So first we're going to look at whether these flowers do in fact differ in time across this area. And then that translates to important reproductive isolation of the gall wasps, because the gall wasps need these new leaves, and that generates reproductive isolation of the insect. So you have this kind of cascading flow, one single trait, bud break, generating reproductive isolation of the plant and generating reproductive isolation of the insects. So first, what happens with the plant? So in this work by Janine Cavender-Bars, she went to a single site. She measured 7 trees of each of the 2 species, and when the first male catkin flowers appeared, and found that Quercus virginiana, which I'll abbreviate as QV, and Quercus geminata, which I'll abbreviate as QG, do emerge at different times. So catkins for Quercus virginiana come out earlier. She found about a 2-week difference. Okay, interesting. One site, 14 trees. We need to find out if it's more common. So we immediately went to all the digital herbaria records across the southeastern United States. We were able to find over 200 or 300 pictures of the vegetative structures sampled across the entire year. And when we marked them as either vegetative only, or if they had the catkins present. And what we found for many more samples distributed across a wide range of time between 1925 and 2011, we found that this same bud break phenomenon and catkins were present earlier with Quercus virginiana and later for Quercus geminata. So that's interesting. Here we found, on average, a 38-day difference, but with some overlap still. We then also monitored bud break in nature. So we didn't trust the pictures. We also went out in nature and measured when we saw new catkins in the field. So I had a graduate student and a postdoc who actually lived in Florida for many months monitoring these populations. And we found lots of variation among trees, but you can see the very dark line up top summarizes the cumulative emergence percentage of the catkins for Quercus virginiana coming out earlier, and the red line documents the average of Quercus geminata, which has catkins emerging earlier. We also followed 100 sapling trees of each species, in a common garden, so grown right next to each other. We monitored bud break as well and found that these trees are very different in their timing, which is critical to the insects. We also measured it in sympatric sites, and even in sympatry, the plants differ in this timing. Very important. So how does this translate to insect timing? How does it generate reproductive isolation among the insects? Well, here we measured emergence of the sexual generation that emerges from these root galls. We did this both in the lab, up top, and using field traps, like yellow sticky traps that just catch insects in nature. In both of them, we found that the Quercus virginiana populations, those that are highlighted in red, orange, and gray, were always earlier, just like their host plants, and the Quercus geminata populations in purple, blue, and green were all later. So in the lab and in the field, we find this cascading difference of bud break influencing both plant reproductive isolation and the reproductive isolation of our insects. Here, about a 2 to 3 week difference, and that's really important for our insects because in nature, as an adult, they live maybe 5 to 6 days. So this is a very big difference to the insect. They cannot survive to see the emergence of the other populations. So what we think we have here is what's called a cascading magic trait, a single trait that's under selection in one trophic level that cascades across trophic levels and causes reproductive isolation at other trophic levels. It causes reproductive isolation for the plants to not generate maladaptive hybrids, and it translates into reproductive isolated insects that feed on plants. We've also spent time thinking about other reproductive barriers that I won't talk about today. Things such as sexual isolation, where individuals from the same host plant mate more often with individuals from that same plant or population than individuals from the alternative host plant. But notice, it's not complete reproductive isolation. They still mate with individuals of the alternative host plant. And habitat isolation. So we do behavioral measurements of habitat preference, letting individuals choose where they want to reside on a cutting of their own host plant or a cutting of the alternative. And on average, especially among the females in gray, the females tend to be on their own host plant. 50-50 would be equal time on both. Females of Quercus virginiana spend more time on QV. Individuals from Quercus geminata spend more time on geminata. So they exhibit preferences for habitat and preferences for who they choose as their mates. All of these barriers, all prezygotic barriers, add up to somewhere around 90 to 95% reduction in gene flow between populations. So they're well along the process of speciation, but that speciation process is not complete. They still exchange genes. Mitochondrial differentiation between populations is still quite low, and we're working now on a genome and a lot of multi-locus nuclear genetic data using RADseq to generate SNP differences to figure out what's going on across the genome. Okay, so I've told you a couple stories about the oak trees and how they generate reproductive isolation of this single species of gall wasp, Belanachnema treaty. But these group of live oaks are attacked by an entire community of gall wasps that attack leaves, stems, petioles, and roots. And so one goal of our lab now is to understand whether the evolutionary processes that generate reproductive isolation in this species are also happening among the entire community. And we're really excited about this. We have individuals that make these very large bud galls here and here. These are two species we expect to see here also on Quercus oleodes in Mexico. They're very interesting because in addition to inducing the tree to grow this tumor, they induce a sugary exudate that attracts ants and other hymenopterans that tend the gall and protect the gall wasp inside among a species-rich group of predatory wasps called parasitoids. So amazing complexity just studying two trees in Florida. It's very fun. But part of this work includes lots of natural history, which is critical to understanding these processes. I want to tell you two really interesting observations and studies that have emerged just from focusing on observing these things in nature and understanding the natural history of these critters. The first is called Tales from the Crypt, a parasitoid that changes emergence behavior of a crypt-forming gall wasp, which we published last year in a couple of journals, Proceedings of the Royal Society and Zootaxa, where we get to describe a new species a species that has been present in the city limits of Houston, the third or fourth biggest city in the United States, that no one's ever seen before. And then the second natural history observation I'll tell you about is called botanical parasitism of an insect by a parasitic vine, which the introduction mentioned. So first, Tales from the Crypt. So first, this project involved lots of collaborators. Kelly Wienersmith was a faculty member at Rice and also a parasitologist, so she was quite helpful in the understanding the relationships between the parasitic wasps and our gall wasps. And then Andrew Forbes is also a great biologist thinking about parasitoid wasps. And then Sean Liu was one of the undergraduates at Rice that helped with this project. And collectively, we worked really hard on this project to describe this new species and its amazing biology. So an overview of the system. First, we'll start off with the plant and the gall wasp. So here you can see the stem of a live oak tree with a couple emergence holes. This signal is always lets me know that one of our gall formers, called the crypt gall wasp, the Setia pallida, is hiding underneath the bark. They don't generate large structures like some of the other gallers that you can see easily, so you have to find emergence holes from previous generations. So that's the first level of the system. And I'll note you'll see a little head peeking out right there. That'll be important in a minute. So first, these ones are only on Quercus geminata, so they're again distributed across the southeastern United States, adapted to very sandy soils. So you'll find these right on the backside of sand dunes, right on the beach, or in the center of Florida, very dry limestone soils. And here are some pictures of both the gall wasp and the crypts they induce inside the stem. So they're just hiding in these little crypts throughout the year. Now back to these holes. When these individuals emerge, they usually leave this nice circular emergence hole. That means the gall wasp successfully emerged. But in the samples we took in this place called Inlet Beach in Florida, we started to notice the cynipid's head peeking out at us, looking at us. And that raised some questions. So we did some dissections. We took this branch and we dissected down into the crypts. Sometimes we found leftovers of the gall wasp and this tiny little white larvae sitting in there too. Sometimes we would just find a different pupa from another parasitic wasp. We had no idea what was going on. So we did just a very small number of dissections. We found some dead parasitoids, but mainly a live parasitoid next to our gall wasp, associated with only these head-plugged holes. The head is in the hole. We never found this in an open hole like this. So only when you found the wasp stuck in the hole did you find this little tiny second insect present. So we let these go on a little longer to see what the biology was going on. And then some of those heads started to have an emergence hole out of the head. So now you have a new hole generated out of the hole the original gall wasp made, which again raised questions. And when we found this new hole in the head, we found this beautiful iridescent wasp, very, very tiny, 1 millimeter, 2 millimeters in length, that started to emerge into our chambers. So we started thinking about this a little further. First, we measured the size of all these emergence holes and counted the number of times we saw these head plugs versus a regular emergence hole. And we measured 3 categories: a regular uninfected Basetia hole, and then what we called an emergence hole by an infected Basetia, where it was either infected but nothing had happened, or infected and a new emergence hole had been found. And what we found is that the infected gall wasp were making a slightly smaller hole. So what appeared to be happening is if you're unaffected, you make your hole, you emerge, and you're fine. If you're infected, you do make a hole, but it's smaller, and then you either get stuck or you just stick your head in the hole and you die. And each time we found this head plug, we always would find this new wasp, which came from the genus Euderus. It's a Eurytomen. So we wanted to ask, is there a fitness benefit to the parasite to causing this behavioral change in its host, which was a gall wasp. So we did an experiment. In the first set of the experiment, or the first level of the experiment, we just simply poked in some of the head plug holes. So we knocked out the hole to see if this maybe changed the abiotic environment, allowing moisture to come in and out, or maybe other parasitoid wasps that might want to come in and go through this barrier. So the first we poked through the holes, and then the second we actually sealed these head plug holes back over with a small thin cutting of the bark of the tree to see if it was actually something about cutting through the bark that was a fitness benefit to the parasite. So what did we find? Well, what we found is first in our controls, so we left some where we just monitored, we had some high degree of emergence of this new parasite, Eudarus. For those that we poked holes in, we found a similar, non-statistically different level of emergence. So they still emerge quite at a high level when you just poke in the hole. There's no abiotic changes that cause a fitness change in our experiment. However, when you covered the holes, either you poked and covered them, or you just covered them alone with the bark, you see a very marked decrease in emergence of the parasitic wasp, suggesting that the parasite needs the gall wasp to cut a hole through the wood to emerge, and that's when it kills it, and then the parasite can then emerge through them. Without that behavioral change in the gall wasp, the parasite remains locked inside this hard, woody galling structure and cannot emerge. So that's interesting. We found this in a single site. How common is this new phenomenon? This is a new species and a new behavior where a parasite is manipulating its host and has not been described in the past. So we started looking geographically across the regions we work in. And once you see it, you cannot not see it. It's everywhere. We found it in Inlet Beach originally, then we found it at some of our other sites across Florida. I found it on the Rice University campus right outside the biology department. I found it at my favorite Mexican food restaurant in Houston called Manuel's. And then I went to the Smithsonian and looked at preserved samples of these galls, and in fact, it was present in the Smithsonian samples. No one had ever noticed it before, but this head plugging phenomenon was present even in these samples that had been in the Smithsonian since 1983. So this is a very common interaction among the live oaks and this gall wasp species that I study across the southeastern United States. And our ongoing work is to find out how common this is across other areas, other plant species, other gall wasp species. We have a little bit of additional evidence from the Smithsonian that this phenomenon has been found in California near Stanford University campus, and some collaborators are looking for it in the Midwest with some very interesting preliminary results. Stay tuned. Okay, so again, this happened because of detailed natural history, of us looking and spending time in nature looking at our organisms. The second discovery is quite interesting as well, and let me tell you a little bit about that. So here you'll see the leaf of Quercus geminata and then the gall of the treaty, this focal species. And now you have a third number. This is a parasitic vine in the genus Casita, which is present in Florida as well. This site, where we first made this observation, is an area where the gall wasp and this parasitic vine are very common. And what we found is a new interaction that no one has ever observed before, where the parasitic plant is attacking a parasitic insect attacking a plant. You can follow that. We looked in the literature, and even though there are thousands of parasitic plants and tens of thousands of galling insects across the planet, no one had ever described this interaction before. So we were very excited about this. And not only were we excited just by the interaction, but also additional observations that it changed the fitness of the gall wasp itself. So here you can see a distribution of gall sizes for, oops, sorry, these are in the wrong order. So this is a distribution of sizes for galls that were not attacked, which you normally see in nature. For those that had the vine attacking them, they only attacked much larger galls. So switch these around. Sorry. In addition, those galls that were attacked by this parasitic vine, we did dissections of them, and in almost half of the galls, there was a dead mummified wasp inside. So not only did it change the gall size, it actually changed the fitness of the gall form. It killed almost half of them. Interestingly, they grew from a larvae all the way to adulthood, which is pretty hard to do in nature alone, but yet once they reached adulthood, we did dissections of these galls, and inside we would just find a dead adult. It never emerged. So the gall is being changed, both in size, and the vine is affecting the fitness of the gall wasp itself. So it's a very interesting interaction, and that interaction seems to have a negative impact on this gall species. We then ask the question, if we see it in this interaction of these two species, how common could you see this in nature? So in this very same patch, these very same oak trees are attacked by many different gall wasps. I showed you the distribution of all those different types of wasps that affect these trees. We again find the vine attacking another species, Caloritis, here forming these, what are called, they're modified root structures called hastoria. They're basically like a suction cup. And they suck onto the side of the tree normally, but here they're sucking onto the side of the gall and changing the gall size of this species. These galls are on average much smaller when they're attacked by the gall former. And then we have other species of gall wasps that induce these bud galls. The vine was not directly attached to the gall, but it formed these very unique double and triple wraps right next to the gall. Again, suggesting that this interaction is a much more common interaction that just we have not observed in nature before. So overall, we found it associated with 5 different gall wasp species on this same host tree. If you can kind of see closely, you see the tree, and then it's just covered in this orange parasitic vine. It's all over. So our goal over the next year is to search at how common this is across the southeastern United States. So again, very important natural history observations that led to new understanding of the system and even new entire trophic interactions. Okay, so back to understanding speciation. We want to think about how speciation may be evolving in all of these systems attacking Quercus virginiana and Quercus jonata. So we first started working on B. treaty, and now I'll show you some similar work on Discocaspis Quercus virens, which is interestingly the one that emits the sugary substance with the ants that tend it. On the left, I'll show you some of the data from B. treaty, and then on the right, I'll show you some data from this new species, Discocaspis. For each of these panels, I use comparisons between populations, where I either have same host comparisons, so comparisons of populations on the same host tree, versus comparisons of populations on different host trees. This same host, different host approach allows us to control for other factors, geography, time, abiotic conditions, while isolating the specific role of ecology, of adaptation to these host plants. What you can see on the right, which I'll focus on right now, Discocaspis, is for things such as the size of the bug measured as height tibia length. Individuals on different host plants tend to be larger than individuals on the same host plant. Just different. And then here you see individuals making different types of galls. So there's more chambers on one species than there are on the other. So you see a greater degree of phenotypic divergence when insects are on the different host plants. So strong differences in adult and gall morphology when you are adapting to different hosts. We also, in parallel, measure habitat preference. So here on the left, I'm showing you that same data I showed you before, where females of one host plant, Quercus virginiana from B. treaty, this gall, tend to be on virginiana, and individuals from Quercus geminata prefer to be on geminata. And the same is true for females that emerge from Distelcaspis. Those females from Quercus virginiana tend to prefer their host plant, and the individuals from the alternative host plant prefer their host plant. So you see some degree of morphological divergence in a second species, and some degree of changes in behavior for habitat preference. Notice none of these are 100%, so we believe that gene flow is still possible, and that we've caught this second species also in the act of diverging, but there's much more work that needs to be done. Overall, our experimental plan is to explore this among all of these different gall formers that we study regularly, and all of these prezygotic carriers and postzygotic carriers. We filled in some of the cells of this table, but we have much more work to do. All right, briefly I want to just describe to you a little bit about the diversity at the next trophic level, the predator level, where we have all of these parasitic wasps and other insects, the natural enemy community that attacks our gall formers. So this is an illustration of just some of the many insects that emerge from a single species of gall former on some of the oaks we work on. Many of these species have not even been described by science as far as we can tell, so we're discovering new species as we are doing this evolutionary biology work. And for Alejandro's lab, I think there's Alorobus, the recording. We also are really excited about this species up here in Bracema, where the females are golden metallic with green heads, and the males are metallic green. So an amazing form of sexual dimorphism, and also what appears to be an undescribed species. So all of this diversity associated with one gall species. And we're interested in this work to think about the process again of sequential speciation. How divergence of an insect onto a new host plant drives divergence of the parasitoid community onto that insect. So this was very clearly documented by a postdoc in my lab when he was working with Jeff Fader on Ragalides fruit flies. There you have a native hawthorn tree and non-native apple trees across North America. The fly has adapted and shifted onto the apple, and the parasitoid community, 3 different reclonid species, all exhibit genetic divergence, behavioral divergence, and morphological divergence, or phenological divergence, timing divergence, associated with divergence of their hosts. So sequential speciation, divergence at one trophic level cascades through the rest of the system. We're doing that in our own system now, starting off with morphology and thinking about timing differences. And this is just at the beginning, but we're already seeing some divergence in morphology of some of our species. And for Tormus, one of the important parasitoid wasps, we see some degree of changing in timing when they emerge, and some degree of habitat preference divergence. So more to be done there. All right, so just even more complex is that I've spent most of my time only thinking about Quercus virginiana and Quercus geminata across the southeastern United States, but there is a group of live oaks that's very well studied, and as you move along the Gulf Coast, you get to Quercus oleoides, which is the Central American live oak species, and there's also another disjunct species, Quercus brundisii, which is an endangered species out in southern Baja. These are all each other's closest relative and represent a unique clade within Quercus, as far as we understand right now. There's a whole area of unknown diversity of oaks right here that I'm very excited to explore in the future. All right, so I've told you a little bit about some of the ecology and behavior. I'm going to tell you a little bit about our genetics. This is going to be quick because I'm getting towards the end of my time. Skip over that. So we're thinking about speciation when we think about the genome, the role the genome plays. We're interested in how the genes involved with speciation are distributed and arrayed across the genome. This is important when you think about divergence with gene flow. So parapatric speciation or sympatric speciation. If you have allopatric speciation, where these genes are doesn't really matter. We described in verbal models with my collaborators Jeff Fader and Patrick Nozzle 4 phases of speciation, where you have direct selection acting on the genome, where we have recombination distance across the genome, and FST between populations. First you have direct selection, then you have some degree of divergence hitchhiking, so local correlated effects of selecting on this part of the genome and pulling other parts of the genome. Eventually, those correlated effects, or linkage disequilibrium, causes genome hitchhiking, where the entire genome starts to be pulled apart, and eventually you have just divergence after speciation has occurred. So we want to start going into some of these systems where we're catching speciation in the act, and look at whether we see these predicted patterns in divergence across the genome relative to the recombination rate. In some modeling done by some of our collaborators, they did some individual-based models and thought about this process. Here you have generation time up the y-axis. They modeled some degree of sympatric divergence and noticed this tipping point. At some point, allele frequency differences are quite small, and then suddenly linkage disequilibrium and divergence across the genome shifts and becomes very, very important, and then divergence among lineages becomes quite quick and rapid. So these are the hypothesis that we're looking for, and we're going to study this in another species, Regulides pulmonella. So Regulides pulmonella is a native fruit fly, a tephritid to North America, including right here in the regions around the EVTM in this area of Mexico. They lay their eggs onto fruits of hawthorn in the genus Crataegus. That baby grows up and eventually eats the fruit and then falls down into the soil, or the fruit falls down to the soil, and they pupate and overwinter in the soil. And then their timing of emergence has to be perfect to match up with the fruiting in the next year of their host plant. So again, phenology and timing is very, very important. They have a single generation per year, and the fruit is the location of where they lay their eggs. It's also the location of where they find their mates. So you have this ancestral host, Crataegus mollis, the native to most of North America, and then as Europeans settled North America, they introduced apples. And because it was an agricultural crop, people watched it regularly. The U.S. Department of Agriculture monitored these populations, and we know exactly when, plus or minus a couple years, when this thing shifted from the native to the non-native apple. It happened around 1850 in the Hudson River Valley of New York State. So since then, in the last 150 or 160 years, these populations have diverged. So there's still a population on hawthorn, there's another population on apple, and chemosensory responses are highly divergent. So individuals from apple only go to apple, individuals from hawthorn only go to hawthorn. And they also have evolved these very strong timing differences. In red, you can see the natural native timing to attack Crataegus. And then these new evolved lineages have shifted about a month earlier, 3 to 4 weeks earlier, to time the fruiting of the apple trees. So we have these important divergence traits, and in about 150 years, these things have evolved into what are almost 2 different species. They still exchange genes to some degree, which makes the genome very important to this system. Other mark recapture studies have documented about a 4 to 6% gross migration rate. That's with actual organisms flying in nature, and genetically the same similar estimate comes out. Consistent allele frequency differences, but about 4% gene flow per generation, which should be so much more. Oh, another Mac PC thing. I'll just describe the experiment we ran. So here we took native populations on Crataegus, and we reared them through 2 conditions. The conditions, the overwintering conditions of Crataegus, which emerge much later, so they have a very short pre-winter time, while it's warm before winter, and then a much longer pre-winter time before for the apple population. So this was the source of divergent selection. We ran these experiments in the laboratory to mimic what happens in nature. We then sequenced the survivors of this lab experiment using RAD-seq, and I'd be happy to talk a little bit about RAD-seq techniques with anybody after this, but in the end, we fragmented the genome, we sequenced it on Illumina, we assembled these fragments, and were able to generate about 33,000 SNPs across the 5 chromosomes of Rhagoletes so that we could look at the allele frequency changes across the genome in a single generation of selection. So what did we find? I'll skip this because I'm running short on time. Here, I illustrate the allele frequency shifts we observed in nature. So lots of loci did not change at all. Some loci shifted in either a positive or negative way, depending on the allele frequency in the ancestral population. And then we used these shifts in our experiment to predict allele frequency differences we observe in nature. And we found, on average, in a single generation of selection, we were able to generate a fairly decent prediction of the genome-wide differentiation between these 2 evolving populations. If we think about it a different way, we can actually map allele frequency change across the genome. So in blue, we see the genomic divergence in our selection experiment, and in red, the genetic divergence among sympatric populations in nature that have been evolving over these 150 years. Amazingly, we were able to recreate much of the genetic differentiation between populations in a single generation of selection in the laboratory. We also looked at this across each of the 5 chromosomes, and on average, we see some repeatability between our selection experiments and the genomic changes we exhibited there versus the genomic changes we observed in nature. If we think about this on average across the genome, we have the average allele frequency here in red of the ancestral population, the hawthorn, the average allele frequency in apple, and then in the dotted line, the shift of the ancestral population reared through apple conditions. So you can see for all loci across the genome, we were able to generate almost 40% of what you observe in nature just through 1 generation of selection in the lab. So the question arises, why? How? First, in this system, linkage disequilibrium is very strong. There are inversions and inversions within inversions, which reduce the degree of recombination. So when you select on one part of the genome in Rhagoletes, you are pulling many different parts. Through some simulations, we were able to estimate that there are about 600 to 700 independent sets across this genome with all the inversions present. Second, these populations have an enormous amount of standing genetic variation. They had the raw material to start this adaptive process. And what's interesting is that raw material of this adaptive process going on in New York and the Midwestern United States, its origin is right here in the EVTM. The populations of Rhagoletes here that have been moving up and down in altitude for things just like timing have traveled through populations and allowed the hawthorns to then adapt to apple in the US. The origin of that genetic variation is right here. And then last, the trait that's under selection, timing, appears to be quite polymorphic. There are many gene regions associated with this trait. So in a study, a microarray study of the expression patterns, hundreds of different loci seem to be up or down regulated during this process of timing within the Rhagoletes flag. So highly polygenic, lots of standing genetic variation, and lots of linkage disequilibrium are what contributed to why these populations were able to shift so quickly in nature, and why we were able to replicate that in a single generation in the lab. So again, I will finish with this quote. Evolution is important to make sense of biology. Ecology is important to make sense of evolution. And I want to thank my hosts and all of the people that contributed to all of the work we've done. It was a team effort, and I'm just here telling you about our team's work. Thank you very much. And if people want to ask questions in Spanish as well, and we can translate, then let's do it either way.
In both the examples you gave us, speciation is not complete, but it's 95% of the way there. Why isn't it complete? Why do they keep going back to maintaining gene flow? There has to be a selective advantage to those hybrids.
Right. So, excellent question. First off, I chose these systems because they exhibit these traits. There are, of course, other examples where speciation has been complete. You know, we look back in macroevolutionary scales, but in microevolutionary scales, we find lots of phenomena like this, where reproductive isolation is not complete. In these systems, hybrids are less fit than either parental form, so there is strong selection against hybrids. But you're right, reproductive isolation is not complete. For the Ragalites fruit fly, they've only had 150 years. So I think if we came back another 10,000 years from now, we might see true biological species. However, the work of many biologists, including Mike Arnold at the University of Georgia, has documented that there's gene flow even after speciation occurs. Rare events of introgression that become important for either lineage. So I view this as a process, a long evolutionary process, and we're catching some of these in the act. That would be my answer.
Thank you.
¿Alguna otra pregunta?
Scott, this is just a comment. I thought that genome and molecular science was taking over and there were no more natural historians. And you have proved that we need to observe more nature, and these interactions can show you not only how species have behaved, but how they are behaving now with all those introductions and all those changes that we're making.
Yes.
As we start agriculture and
Yes, no, I think you touched on an important challenge of the modern biologist, is that you have to be able to do everything, which is impossible. So all of this work represents collaborations. My PhD was spent catching beetles in Canada and northern US. I was not doing any genomics, but I started to collaborate with Greg Ragland, who was doing the genomics and transcriptomics of timing differences, and Jeff Fader and Patrick Noswell. So I had to build teams to address these important topics. In no way am I a genomics person would not call me a genomicist. A behavioral person probably would not call me a behavioral biologist. Right? So it's important that the modern biologist needs to link all these things, but you can't do it yourself. So building collaborations has been important for my work to be able to do this. I'm probably more on the natural history side of things. But I understand the importance of the genome and understanding these events. Yeah, good point.
Could you show examples of specialized, specific species of insects in specific species of plants? How do you think this model would stand in more generalistic species?
Excellent question. There are certainly many generalist insects, but on average, most herbivorous insects are specialized. And that gets at another question that my lab does address that I like to think about, which is why? Why do insects specialize? Which is probably what promotes the speciation process itself. For the parasitic wasps that I study, the historical literature reports many of them to be generalists. When I look very closely at the systems I work on, their behavior, their morphology, or the genetics, I'm actually learning that things that were described as generalists are not exactly that generalist, they're specialists. There are still generalists in nature. And then it gets at another interesting topic, which is how you quantify generalism versus specialism. The leaf beetles, which I did not have time to talk about, that's one species that's oligophagus. It feeds on one species of willow, one species of maple, one species of alder, and rejects a sister species of willow, a sister species of maple, a sister species of alder. So I actually think there you might call it a generalist, but it's finding something in the chemistry of those three plants, or the timing of those three plants, that actually overlaps. So even those things that are more generalist might actually not be so generalist. They may be specializing on something that we, humans don't see, but the beetles see. So I think that could be going on. So there's a spectrum of specialization. Most herbivorous insects are specialists, and we think about what drives that. And then we also think about how we measure specialization. We're working on that with a big data set of chrysomelids right now, using the agricultural literature and entomological literature to understand patterns of specialization. So, great question.
Bueno, tal vez tenemos tiempo para una pregunta más, corta. Si no, pues, thank you very much for this talk. Los invitamos a ponerse en contacto con el doctor Iván en el momento del café para que sigan platicando con él un poquito más. Muchas gracias por su asistencia.